we will just start. So today we'll talk about plant transgenesis. Let's talk about first what would you do with a plant? Like what would you do? Why would you do plant transgenesis? What do you guys think?
Increase yield.
Increase yield. How would you do that? What could you do?
Something that grows more on. Okay, so kind of like the fish example you were talking about the other day. Find something that grows faster.
Yeah, you could make something that grows faster. So if the catch is that you only have a particular season of good weather, maybe you want to increase your yield. Maybe you want your crops to grow faster. So you could make transgenes that potentially make it grow faster. In an animal like the catfish, they were talking about putting in a growth hormone. You could find a similar kind of a thing in a plant that makes it grow faster. So you could do growth rate increase. You could also look for, like, what are the other issues that people who do crops and plants are dealing with? Diseases. And then also like nutrition, so like drought. So you might want to make a strain that's drought resistant or disease resistant. And there's all kinds of ways that you could do that through insertion of transgenes or breeding or deletion of genes. You might also want to encode insect protection. So we'll have a whole series of lectures on BT. And the principle of BT is they found bacteria that makes a toxin that kills insects. And they took the toxin and they put it in a plant. Okay. And then whenever an insect eats that plant, the insect dies. So you can encode insect protection in transgenic plants. Another way you might be able to increase the yield is, right, like plants have chloroplasts. What are chloroplasts?
They photosynthesize.
Right, their job is photosynthesis. So they take energy from the sun and they convert that into basically the energy in the plant. And there are a lot of people who study, so photosynthesis, you've learned in basic biology, there's this electron transport chain in the chloroplast, and it passes electrons through and collects the energy. So there's a lot of people who look at, is it possible to make the electron transport chain more efficient? So can we increase the efficiency of basically the plant itself? Is there a way to increase the efficiency of how it can take in energy of the sun? And then in that sense, they would convert it to more energy. And if you eat it, then you would have a more nutritious plant. So a lot of people who study that. So there's a lot of ways that we can think about of how to change plants in a way that might be beneficial for humans or agriculture. Okay, so the next question is, how do you do that? How you change the plant? What are all the ways that we can think of? Breeding, right? Like classical breeding. This is what when you hear biotechnology lectures and people say, well, none of this technology is really any new. Like we've been breeding plants for tens of thousands of years, since 10,000 years ago when humans had the agricultural revolution. We've been breeding plants to give us the traits that we want. So we're imposing selection on them to do what we want. And of course, by doing that, we're changing the genes. So we've always been changing the genes. It's just now we have more targeted, specific ways of doing that. So breeding would be a classical way of shifting allele preferences. So what are alleles? Right. So if you have geneX and another copy of geneX, so X1 and X2, you might have one particular mutation in X2. These are different alleles of the same gene. And it might be that this particular X2 gene gives us drought resistance. And so we want to select in the next generation a homozygous plant that has 2 copies of X2, right? And we can do that through breeding, just classical Mendelian genetics. So breeding is one way we can do this. A lot of people also work on hybrids. So what's a hybrid?
Mixing 2 species.
Right, mixing species. But how is that possible, right? If the definition of species is that they can't breed with each other. Is that the definition of species?
Well, the offspring hybrids can't breed.
Are usually infertile. Right, very good point. So what we can do is we can take, say you have wheat species 1 wheat species 1 or catfish species 1, and you can take that species, you take a male, and then you have a second species, wheat species 2. I guess in plants, I'm not sure if you do the male, the female, but imagine it's catfish, right? You take a female from a different species, you mate them together. This is the F0 cross that creates the F1 generation. And the F1 is going to be a mix, like a donkey. It's going to be a mix of the two different species. And so you're going to get beneficial traits from species 1, and you're going to get beneficial traits from species 2, and you're going to mix them in this F1 with the catch that usually this F1 is infertile. So the catch with this is the companies like Monsanto, they have to prepare these hybrid seeds. So they have to grow species 2 and species 1 and then prepare them and breed them to create the seeds that people then plant for the F1. Okay, so that would be one means of doing genetic modifications in plants by creating hybrids. Okay, so we have breeding, hybrids. What are other ways we can do transgenic, or I guess not transgenic, but alterations in plant biology? Transgenics, right. So there's different thinking on transgenics. When you hear transgenics, usually that's inserting foreign DNA. So you're usually inserting something, ORF, or a gene that wasn't there before. You're putting that in. The other way often is in terms of you hear gene editing, or you could do classical mutagenesis. Usually these are used for knockouts. So you're knocking a gene out. So just like in yeast, we talked about making deletion of UR3. In plants, you might want to knock out a gene. But when we say transgenics, it's usually adding something in. And when we say gene editing or mutagenesis, it's usually knocking something out. And these actually have different regulatory mechanisms with the government, and we'll talk about that. But first, let's talk about the transgenics. So we'll talk about how to make transgenics. Okay, how would we do that? You guys have heard the lecture before in the previous biotechnology class. What's the most common way that they make transgenic plants? Agrobacterium. So we'll focus a lot today on agrobacterium. So agrobacterium is still cutting edge, still used every single day. And I called Monsanto before I planned this class, and I talked to some people there, and I was basically like, well, what do you want biotechnology students to know? And they said agrobacterium. It's still used every single day. So if you hear all the excitement about genome editing or CRISPR stuff, that's often we'll use that in very specific contexts where it has an advantage over other technologies. But there's still technologies that were developed 20, 30 years ago that are still used every day, and they're still cutting edge, and there's still a lot of stuff that we don't understand. So when Dr. Con Wang came and talked to you guys, one of her specialties was agrobacterium and how agrobacterium is used to make transgenic plants. So agrobacterium is gram negative. What's that mean? No cell wall. But there's usually a catch. It kind of usually means that there's kind of like a tiny cell wall. So in gram-positive bacteria, you have a huge thick cell wall and an inner membrane. That's positive. In gram-negative, usually the bacteria have an inner membrane. They have a tiny thin cell wall, usually, and then they have an outer membrane. So the gram-negatives have two membranes, outer, inner, and a tiny cell wall. Sometimes they have no cell wall, if you're talking like mycoplasma. But like Wolbachia, which I study, agrobacterium, they have kind of like a tiny thin cell wall. Okay, so it's gram-negative. It lives in the soil. You find it out in nature. And it has this technique that it seeks out plant wounds. So how would it do that?
Whenever plants are wounded, they have a wounding response that releases hormones.
Right, they release chemicals. So if you have a plant, I'll just say a leaf, but maybe it's the root. and you get an injury, there's going to be chemicals that are released into the environment, like sugars and phenolic compounds. Okay. And Agro, if it's in the soil or if it's on the thing, it can sense that, and then it can start making its way, and it seeks out and it finds those plant wounds. Okay. So it finds plant wounds, it senses them in the environment, and within Agro, so if this is Agro, it's got specialized machinery for virulence in plants. So from what we've been taught, what I've taught you guys about bacteria, where do you think you would find these virulence factors? On plasmids, right? Like I talked about that in the basic biology of plasmids is what do you often find on plasmids? You often find things on plasmids that are virulence factors that help it invade in very specific contexts. So you might envision that Agro is usually living in the soil. It's not usually replicating in a plant, but in that off chance that it finds the plant. So if this is the chromosome, it's got a little plasmid, and the plasmid has a specific name. It's called the Ti-plasmid. Do we know what that means?
Like the Ti-84.
No. What does it do when it infects a plant?
It causes crown gall disease.
Right? And gall is what kind of a structure, I guess? It's a tumor. And so this plasmid is called the tumor-inducing plasmid. Okay? And its job is to induce tumors in plants. And why does the Agro want to induce a tumor in a plant? Yeah, it basically is like forcing the plant, manipulating the plant to create a little environment, a little microcosm that this agrobacterium can replicate in and basically steal nutrients from the plant in favor of reproducing the bacteria. So it's got this whole Ti-plasmid whose job is programmed to induce tumors in the plant. So let's look at the Ti-plasmid. I have a picture of it right here. So here's a diagram of the Ti-plasmid. And what are some, like, what's this? This is the origin. And this origin is specific for Agro. Okay? It's got virulence genes. So on this section, you would see a bunch of these genes, virulence genes. And the virulence genes are abbreviated VIR. So you'll see like VIR-D4, VIR-E2, VIR-D8. These are all quote unquote virulence genes. Their job is to induce the tumor or cause basically stress in the plant. Okay? And then there's this tDNA section, which is what Con Wang studied. So we're going to talk about that. And there's a T left and a T right. Okay? Which is kind of like when we talked about transposons. When we talked about transposons, I talked about the helper and the donor plasmid. And the helper encodes what? The actual enzyme, the transposase. And the transposase protein recognizes what? Right, it recognizes consensus sequences. And you often put these on the left and right of your geneX so that your transposase will get made and grab these regions of the DNA and cut out your geneX and insert it onto a chromosome. Okay, so the Ti-plasmid is kind of the same way. If you look at its structure, it's got these sections of DNA, which are recognition sites on the left and the right of this tDNA sequence. And the virulence genes encode proteins very similar to transposase, which can recognize these sequences, okay? And they grab it, they literally hold it. The VIR-E2 protein, we'll get to the protein in a second, but one of these proteins' job is to grab the DNA, okay? And then what happens is, if you're into bacteria, you got the Ti-plasmid. So here's another question. So you got the Ti-plasmid. Do you want the Ti-plasmid always expressing the virulence genes?
No.
Okay, so how would you program it to only express them when you want them? How would you program it?
Maybe it only activates in the presence of, like when it's inside the plant already.
Inside the plant, but that might even be too late. You got to have it in. You got to have a mechanism for it to invade the plant. You know,
The pheromone.
right? So you might have a promoter that only turns on in the presence of the plant wound pheromones, right? And who knows what those might be? Maybe that's a specific chemical. Maybe that's a specific protein. Maybe that's a specific sugar. But you can build proteins that recognize these things, which are transcription factors, and then you build a promoter that only binds, just like the air promoter, the lac promoter, we talked about those. You only build a promoter that turns on when that chemical is around, okay? So then the virulence genes turn on, and then the virulence process starts happening. Okay, and then what happens, looking at my notes, we already talked about how it induces gall formation. What a next key step is the Agro. So let's look at the Agro. It's gram-negative, so it's got the thin cell wall. What's the next step that has to happen? You got this Ti-plasmid. Let's say you got the chemicals, they came in, it turned on the plasmid. What's the next thing that has to happen? Let's say it's already at the plant site.
It has to insert its DNA.
Right, and the DNA has to get like out of the cell, right? So you have to have, I've talked about this before, you have to have a programmed machine or way of getting this plasmid out and into the plant cell. That doesn't just happen. The cell is programmed to keep stuff in or be very, very selective of what it's letting in and what it's letting out. Because if you just let everything in a membrane, you die, right? So there's a machine in the membranes called the T4SS, which stands for what? Right, SS, whenever you see that, that's secretion system. And there's a whole bunch of secretion systems in bacteria. You can imagine there's a type 1, a type 2, a type 3, a type 4, type 5, type 6. So Agro uses the type 4 secretion system. And oftentimes you'll see the 4 in Roman numerals, type 4. Okay? So you probably heard of type 3. The type 3 is basically like a syringe. And bacteria will make like a syringe and they'll literally like stab and pump stuff out. Okay? Type 4 is, they say it's quote unquote ancestrally derived from conjugation. What's conjugation? We've talked about that. Right. So conjugation is bacterial sex. So bacteria have ways of mating with each other and exchanging DNA, and often what they're exchanging are plasmids. So in conjugation, two bacteria will find each other. I'll draw this other bacteria further away. Two bacteria find each other, and conjugation machinery will turn on, and the conjugation machinery builds like a little tube made of proteins, conjugation proteins, okay? And it connects the two bacteria, and then information can pass back and forth. So genetic information. So this is bacterial sex, this is conjugation. Well, it turns out that one of these conjugation systems has evolved in ancient bacteria to become a secretion system. And in Agro, they have evolved that system to basically inject plants with a bolus of the tDNA. Okay? So that's the type 4 secretion system. And I'll just say that I won't spend too much time talking about the structure of the type 4 secretion system, but I will say that if you have the bacterial cell, you have to imagine that the T4SS has to span three things. It has to span the inner membrane, the cell wall, the thin cell wall, and then the outer membrane. It's got to span the whole thing, okay? It's got to create a channel. And I'll also say that this type 4 secretion system is like a machine, which has a bunch of different subunits. So it's a protein complex. What's that mean? Right, there's multiple subunits of proteins that make up a full complex, a full machine, okay? So that's why when you see like VIR-D4, VIR-D4 might be the protein that holds it right there. VIR-D6 might be the protein that couples and looks for substrates in the cytoplasm, and then it grabs them and then feeds them in. And then there might be another protein, VIR-D8 or something, that will take energy from ATP to create like a pumping mechanism that pumps stuff out. Okay, so I'm basically telling you that there's different subunits, different components of this machine, and they're usually all VIR-X, VIR-Y, and they're usually organized in an operon. So what's an operon again?
It's like a series of genes that have not the same function, but
They work together.
They work together, right? Like, so when you encode the genetic information that builds this type 4 secretion system machine, you usually encode it in an operon, and it doesn't make sense to turn on only one component of the car or the truck, like when you need them all. So they usually will have one promoter, and maybe this promoter is even shared in expression with the virulence genes in the Ti-plasmid. Maybe what you're really turning on when the cell senses the wounding of the plant is you're turning on this machinery to build this secretion system, okay? And then that's why they're labeled VIR-D4, and then the next gene will be VIR-D5, and then the next gene will be VIR-D6, and then VIR-D7, whatever. You know what I mean? Up to N. Okay? So these things are usually organized in operons, and the secretion system has to turn on at the same point when the agro wants to create the cancerous tumor. Okay. So then the basic outcome is the agro injects the tDNA with some proteins. One of these proteins is VIR-D2. One of these proteins is VIR-E2. And VIR-D2's job is to hold on to the tDNA and to bring in this VIR-E2 protein. We don't quite know the function of the VIR-E2, but I would just say, here's what you want to remember, is it's kind of like a transposon in a sense that there's proteins that are programmed to hold on to the tDNA. So this VIR-D2 brings it in, and then you're not done yet. If you're sitting in the cytoplasm, what's going to be the outcome of that?
It might get cut.
Might get cut, it might get degraded. It's not going to integrate into the DNA. Why?
It has to be integrated. It has to be machinery to put that together.
Where's the DNA? In the eukaryotic cell. So here we're talking about, this is why I taught you procs versus eucs. This one is a plant cell. Because eucs have what?
So the nucleus is going to be membrane out. So
Yeah, there's a wall called the nuclear envelope, which holds in the nucleus, and all of the DNA for the plant is in the nucleus. So if the goal of the agrobacterium is to change the DNA of the plant, to cause it to change its functionality and do different things and induce a gall, it's got to get that tDNA into the nucleus. It can't just sit in the cytoplasm. So it not only has to get secreted out of the bacteria, it's got to get into the cytoplasm of the plant, and then it's got to get through the nuclear pore complex, which is a hole in the nucleus, the nuclear envelope, to get in to find the DNA, okay? So one of the other jobs of VIR-D2 is to couple with the machinery that imports things into the nucleus, and it gets imported so that your tDNA can finally integrate into the genome. And then once you have that integration into the genome, you're starting to now change the expression of the plant, of what factors the plant is making. You might have it expressing stuff from that tDNA, which causes it to form this cancerous gall, which helps other agros. Does that make sense? So that's the basic goal. Okay, so let's talk about the tDNA. So the tDNA has these sequences, T left, T right. You could also think of this as 5', 3'. So now my question is, it's obvious, right? Like, how would we hack the system to insert a gene that we want? What do we need to do?
You could remove the gene that's there and put in your own.
Yeah, you cut that out and you put geneX. Okay? Now what Agro is inserting is not the tDNA, it's inserting the geneX that you inserted there. And if you built in a little promoter, a plant promoter, okay, then that means that plant is now going to be transgenically expressing your geneX. So that's in very simple form. That's all that needs to happen. Okay, and then all that needs to happen is, so this would be an integration. If we want to integrate something geneX into the DNA, all you need to do then is grow some soup of Agrobacterium, and you literally just like grow like a little soup, and then you can take your plant and you can like dip your plant in the soup, and if there's a little wound there, it's going to pick up the Agro, and then you're going to get this transgenic process to start going. So transgenics with Agro is very, very easy. That's why we like doing it. Okay, let's talk about some of the caveats and the catches with this system. The first thing to note is that we don't quite understand yet the mechanism of integration. And one of the downsides of it, it could be downside or positive side in certain contexts, is that it's random. So I keep making comparisons to the transposon because the transposon insertion is at a random location, somewhere, anywhere in the genome, okay? In Agro, the tDNA inserts into the plant genome at random sites. It's not site-specific. It does not equal site-specific. You have no control over where it's being inserted.
Dr. Becker,
Yeah.
this bacterium infects like the stem near the roots.
Okay.
So how does it change the seeds?
How does it change the seed? Well, you could like dip the seed in there. You know what I'm saying?
Okay.
Or no?
Or you could, I was thinking you could like propagate from.
Right. That's why we had Dr. Bowen give you the micropropagation lecture, is because that's what people will do is they will do a transgenic region on a plant, and then they can like cut that piece of that plant off, and then they can like micropropagate that and select for the specific thing that you want to insert.
So it never has to enter the genome. So it can just.
Well, you can do it with seeds. They also do it with seeds, and then they can like plant the seeds and the seeds that survive. So I'll actually talk about that. I think you can do it. You can do it with not only seeds, you could do it with like a leaf, and then you could micropropagate the leaf, or you could do it with the root, you can micropropagate the root. Like you can do it with different regions of the plant, is my interpretation. Okay, but it's random. Okay, so I was going to actually, the next point, which actually segues into what Wilson said, is because it's random insertion, what can you imagine one of the uses of random insertion would be?
Maybe the best place for the gene to go is something you would expect. Like it's
Well, what would be like an actual, like, what do scientists often use the randomness of insertions to, like, what do they use that to make? Maybe that's a bad question. So, for instance, if you take a transposon that inserts, oftentimes what we make is we make a whole bunch of different lines that where the transposon has inserted into different regions to break different genes, right? So you could make a knockout library. So one of the things that they've done with Agro is they've taken Arabidopsis seeds, okay? So Arabidopsis is a plant, and they've taken the seeds, and they've made 60,000 Agro insertions of the tDNA at 60,000 sites, and each of these 60,000 sites is a broken gene. So they have a whole knockout library of Arabidopsis where if you want to study, if you're looking at some gene from Arabidopsis and you need to study it, but nobody's studied it before, there's a 75% chance that there's a library that you can pull out of a seed that already has that gene knocked out. Does that make sense? So like people have used this random insertion process to just cover insertions throughout the genome and then isolate individual lines so they can keep the seeds of this knockout library. Does that make sense? So one of the advantages in that it's random is you can use it to basically shoot shotguns at the whole genome, and you just isolate where it hit, and you can isolate different lines that have different knockouts. Okay, so that's one of the things that they've done with Arabidopsis. Okay, so another question that we don't quite understand this is we still don't quite know how the tDNA integrates. And by that I mean, like, is it homologous recombination? Is it some unknown mechanism? How is it actually like taking? So if this is plant chromosome, plant, how is it actually getting into the genome? We don't quite know that yet. The best hypothesis is that there's some kind of taking advantage of non-homologous end joining. So we've talked about that a little bit before, and I always keep saying, well, at the end of the semester, we'll talk about CRISPR, and this is more related to how CRISPR is working. But non-homologous end joining is, what is that?
You're putting together pieces of DNA that should match, that don't match.
Well, why does the cell have this process? So you're right, it's when the cell takes two pieces of broken DNA and it just smashes them together. That's non-homologous end joining in its simplest form. Why does the cell have a mechanism to do that? Yeah, double-stranded breaks are lethal, like for the cell. It needs to repair. So it's a repair mechanism. It's a eukaryotic, well, I think it's prokaryotic too, but it's a repair mechanism of DNA. Okay? And so it's thought that when the Agro starts injecting its stuff, the tDNA finds a spot where there's a double-stranded break, and it kind of wedges itself between, and then you have two instances of non-homologous end joining where it smashes the DNA together, and then you get this integration. But we don't know. This is one of the models. This has not been proven yet. So if you read the most recent review, I read a 2017 review, they still don't quite know the mechanism of the integration. Okay, but it's safe. All you guys need to know is that it's random, and it seems to mimic non-homologous end joining, which means it just pops in.
Not to go too deep on that, but wouldn't that be a very non-efficient method to like?
Well,
how common are just random double strand breaks with them?
Well, one of the ideas is that the Agro might have a mechanism of inducing double-stranded breaks. You know what I'm saying? So yeah, like, and I'll also say, like, double-stranded breaks are very, very common. That's why we have repair mechanisms, because like UV light or this, like you might encounter a mutagen, like we're always getting breaks in our DNA. That's why we have these repair processes, and that's why if you knock out genes from these repair processes, you die because you can't repair your DNA. So it happens quite a bit. But you're right in a sense that it might not be as efficient as the bacteria wants. So the bacteria might actually encode its own means of breaking the DNA, which could be like a nuclease, or it could be some kind of chemical stress. But again, I'll just say unknown at this point. We don't quite. There's still a lot of room for research. Okay. So let me ask a question. If we have CRISPR-Cas9, CRISPR-Cas9 can do the same thing. You can cut the DNA, and then you can rely on, you can direct it to homology-directed repair and give it a template that matches, and you can get recombination of geneX into your plant genome with CRISPR-Cas9. So let me ask a question. I'm telling you, you can insert genes with CRISPR. You can do that. You can also insert genes with Agro. Why would you choose one or the other? Why would we still be using Agro today? Any thoughts?
Just be easier. The cell already has a lot of those mechanisms integrated.
I don't know if it's quote unquote easier. There's a better reason. One of the reasons is Agro is very, very good. So Agro involves transferring a plasmid. Okay. And this plasmid is quite big. It's like 20,000 base pairs. Okay. Yes. Okay. Very, very good. So you can make massive insertions, like massive, like 150,000 base pair insertions into the plant. Okay. I think the record I read was 150,000. With CRISPR, you cannot insert that big of a size of genes. And let's imagine we're going to change a plant. Let's imagine we need to add in a new metabolic pathway. Are we going to be so for instance, like golden rice, okay? In golden rice, they introduced a whole metabolic pathway to synthesize a particular vitamin, okay? Do you think that's going to be one gene or 10 genes? It's going to be multiple genes. So it's very rare that we want to insert just one thing into the plant. Oftentimes we want to insert a whole pathway, okay? And if you're going to do that with CRISPR, you're going to have to do CRISPR over and over and over and over and over again. If you do it with Agro, you could synthesize the whole construct and you could insert that 150,000 base pairs straight into the genome at one shot. Does that make sense? So if you're thinking in terms of size, okay, if you want to insert stuff that's large, Agro is the way to go, okay? So that's one reason why Agro is still used today. Okay. So let's talk about kind of like how you do it, like how you build the stuff to do it. So here's a question. We talked about conceptually, we can just take the Ti-plasmid, and in theory, you got the left, the right, you could just insert geneX right there. But let me ask you, like, how would you do that? How would you make that insertion? What are you going to have to do?
You have to do PCR.
Okay, so let's say step 1, you PCR geneX. Then what?
You'd have to be running like cultures of agrobacterium.
Cultures of Agro, okay. And one question is, what's going to be the copy number of this Ti-plasmid? Going to be high or low? It might depend. Yeah, like it's not necessarily going to be a great plasmid for engineering stuff. Like, what have I taught you about engineering and plasmids? Is it better to work with small things or large things? Right. So it's a lot easier to clone geneX, even though I just told you it's good for great big things. But it's easier to clone geneX into a tiny plasmid. So are you going to want to work with a plasmid that's already 20,000 base pairs? And are you going to build it in Agro or are you going to build it in E. coli? You're probably going to build it in E. coli. So is that origin going to even work in E. coli? Probably not. No. So oftentimes what people do is they will take a plasmid, a little plasmid. So let's say it has an origin PBR322, which is a high copy. I think it's like 600 copies in E. coli. Okay. And they will insert geneX onto the PBR plasmid in E. coli. Okay. And then we can make homology regions that match. So let's say you make a region that matches here and a region that matches here. And I'll make this the endogenous Ti plasmid. Okay. So you make homology regions that match here and here, right? We can build that. I taught you how you would build that. Okay. And then this plasmid, let's compare the origins of this plasmid. This plasmid is PBR322. Let's say you also insert, let me make this more complicated. Let's do 2 genes. Let's do geneX that you want, and then let's also do kanamycin cassette. Okay? Now, let me tell you what's going to happen if we take Agro, which already has the Ti-plasmid, and when we transform it with this plasmid, with XCAN. Let's look at the origin. So I just told you This origin is probably not going to replicate in E. coli. Is PBR322 going to replicate in Agro? No. Okay. So this is a suicide plasmid. Again, very similar to transposon. You give it something that it's not going to keep replicating in the Agro. And then you can start selecting, if you have the regions that match, what might happen? They can swap. They can do recombination. So you can swap this. Then what's yielded is, in some infrequent population, you have Agro that has a Ti-plasmid, which now has been swapped with geneX and kanamycin, and your PBR322 plasmid now has that tDNA. Okay? As this replicates, if you add kanamycin, the only cells that are going to survive are the cells that swapped that kanamycin into the plasmid that has the origin that can replicate. Does that make sense? This one's just going to get lost because it's not going to replicate. So then what you select for is you select for the bacteria that swapped out your plasmid, swapped out your X and your kanamycin, and then that is what you dip your plants in. Then that gets transferred, that gets sent to the nucleus, and then your geneX and the kanamycin cassette can insert into the eukaryotic, the plant, chromosome. Does that make sense? Yeah.
So that region of DNA.
Which region? This?
Yeah, the tDNA, where it's been replaced with those genes.
Yes.
The bacterium uses those. I thought they were just meant to be cut out and inserted.
Well, what do you mean by uses them? So, like, are they being expressed? It would depend on the promoter you put in. But, like, uses them, like, what's the same are the T left and the T right. So the Vir D, whatever it was, the protein that grabs those sites, it can't tell the difference between what's been swapped out because the regions that it grabs here and here are still there. Does that make sense?
Like kanamycin resistance, that whole like machineries.
Oh, I see what you're saying. Yes, when you select for, yeah, so the kanamycin gene is going to have a plasmid that this gene will be on. And if it's not on, they'll die, right? So yes, you're correct. But your geneX, you might have programmed that with a plant promoter, and maybe geneX is not on yet until it gets into the plant. But this is good that we're talking about the kanamycin, because what's interesting is when that plant nucleus, when that kanamycin cassette, so you have X plus kanamycin, gets inserted into the plant nucleus, we can actually select for the plant, select for the transgenic plant based on the kanamycin cassette. So we can grow the plant in plus can media or whatever the micropropagation wax mixture, whatever, you know, that media that Dr. Bowen made. You can put kanamycin in that and you can select for plants. So my question is, how does that work? Because kanamycin is an antibiotic, right? How can you use it to select for a plant that has a kanamycin resistance gene insert?
Maybe put him back.
How does kanamycin work?
Well, you would need it to have, your plant needs to have the resistance to it, right? So
Not necessarily. So, I mean, how does the mechanism of kanamycin work? So kanamycin is a chemical that will inhibit ribosomes. Okay. And interestingly, so let me ask you another question. Okay, but it only inhibits bacterial ribosomes. So let me ask you another question. Is there anything in the plant that looks like bacteria? Yes, the chloroplasts and mitochondria are ancestrally bacteria. So the chloroplasts actually have ribosomes. They have their own ribosomes. And kanamycin actually also inhibits the ribosomes of the chloroplast. So the reason you can select for plants and bacteria with kanamycin or its similar chemicals is because you get the same thing happening in the chloroplasts, okay? So we can select for the plants, transgenic plants, based on the kanamycin cassette. Okay, I'm going to move to the next section quick because we only have 5 minutes left. So that's agro transformation. Now you kind of know the gist of it. There's other ways to transform plants. And I show you this because this is just the most awesome story. And I think I put on the thing that sometimes crazy, dumb ideas change the world. So there's the particle bombardment. And what's particle bombardment?
Gene gun.
The gene gun. Okay. And when I was reading about this, this is the most crazy thing. So the first iteration of the gene gun was these scientists took an air pistol, like an air pistol you buy at Walmart, okay? And they made tungsten pellets, beads, and they coated the beads with DNA. So they had little beads, and they had a gun, literally like a gun from Walmart. They loaded these DNA-coated beads, and they shot an onion, okay? And the DNA on there was a reporter. What's that mean?
It'll show up.
It's a gene that'll get turned on. Let's say it's GFP, or let's say it's LAC-Z. It'll show you something if it gets inserted into the genome. So they literally just took a tungsten bullet and shot it at onions. And then they looked at the onions, and they found that some of the onions were picking up the DNA that they shot in there. So that was the first like proof of concept of particle bombardment, where you could shoot a plant and shoot in DNA. So I just thought that was the coolest thing. So now it's a little bit more robust. Like you have like a whole apparatus. You set like a little culture of plant tissues, and then you like press a button and it will like bombard with DNA coated particles. So not like this anymore. But you can do that. You can use a gene gun to also insert. And the reason I tell you this is because, let me ask a question. So now we've talked about, you can do CRISPR insertions, you can do Agro insertions, you can do gene gun insertions. Okay, and gene gun is still used today as well, very, very commonly. And there's a reason for that. Gene gun is particularly good at something. And it's particularly good at inserting into the chloroplasts. So there's cases where if you have a plant cell or even an animal cell, and you're talking mitochondria or chloroplasts, the chloroplasts are basically, or the mitochondria, are almost like little tiny organisms that live inside of you. I mean, they're not, they can't live by themselves, but they were ancestrally were a different organism, okay? And they replicate, they have their own DNA, they replicate, they have their own genomes. And there's some cases where you might want to insert into a chloroplast genome. So how many chloroplasts are in a plant cell? Is it one or many? Many. So if you want something to be very, very, very robustly expressed, it might be in your interest to insert it into the chloroplast. Or if you want to engineer a plant like we started in the beginning to have a more efficient electron transport chain, maybe you want that gene straight in the chloroplast, right? So the gene gun is particularly good at getting genes in the chloroplast. So that's why people use that today. But it's also itself is also random insertion. So really, CRISPR-Cas9 is the best means of doing site-specific insertion. So if you want to do a specific site, you use CRISPR, but you can only insert small things. If you want to insert either randomly or really, really big stuff, you go to Agro, or if you want to insert into the chloroplast, you can use the gene gun. Okay, so let's just finish with talking about regulation, because this is kind of funny. So regulation of how does the USDA or the government think about these different transgenic technologies? So if you insert any foreign DNA, that's a GMO. That is special regulation based on GMO. It's a transgenic. Okay? If you use CRISPR-Cas9 for a knockout, you're not adding anything new there. Okay? So they do not consider that a GMO, okay? And if you use gene gun to do a knockout and you don't add in anything new, that's not a GMO. So even though you can manipulate or modify the DNA, it's not considered genetically modified unless you're adding something in. And that's that. Any questions? Yeah.
Could the gene gun be site specific if you're just really active?
I would never say I don't like to say things are impossible because maybe, who knows, maybe we could like make like a micro gun. But and usually what you're doing is you're just kind of shotgunning in a bunch of DNA and just kind of hoping it integrates. So that's my answer. That's my final answer. All right, that's it. So next week, back to normal, lab on Monday. Quiz on Monday. Thank you, Dr. Liu, for attending.
Very nice.